All right, good morning. For those that don't know me, my name is Danielle McLeod-Henning. And so the next 4 projects are going to be presentations from my portfolio area at NIJ, encompassing forensic anthropology and forensic pathology. And without further ado, we will go ahead and get started with our first presenter, which is Catherine Wysensi from Clemson University. So Katie, if you want to go ahead and jump up. Thank you.
All right. Can you hear me okay? Good. And I found the thing. All right. Thanks so much for inviting me to talk today. I'm excited about talking about this project that we've been working on for the last couple years on improving PMI estimations through mass collaboration and machine learning. I want to acknowledge my collaborators. They are the people that have made this project possible. Christina Tika has been in charge of organizing and collecting data from various institutions throughout the country. Patricia does our spatial analysis, and Carl and Hudson are the data scientists working on the project. This project is part of a 3-year funded project, and we're about halfway through. So I'm excited to share our progress and updates of the project so far and talk about next steps and places that we're going. PMI is often one of the first questions investigators want to know when human remains are found, and it's the thing I feel least comfortable in providing, given the methods that are currently available. Methods for estimating things like ancestry and sex were developed on large reference data sets that are forensically and geographically relevant in data that's continually updated in the forensic data bank. It's really this model of the forensic data bank and 4-disk and the power of mass collaboration to answer questions important to the forensic community that was the impetus for the current research. I want to start by giving some context on the current state of the field. This graph here shows the published literature from 1989 to 2021 that used human models to estimate or test PMI methods. Each bar on the graph represents a published study using humans in an experimental study of PMI. And the height of each bar represents the number of samples used in each study. So you see that over the last 30 years, about 5,000 individuals have been used to develop methods related to PMI. Of those studies, about half of them were related to the first hours after death, with things like vitreous humor, cooling, and livor mortis. Therefore, most of what we have documented on human decomposition in the days, weeks, and months after death is based on observations from about 2,500 individuals, with a median sample size of these studies of 18 subjects. This is a small number, given that we know that decomposition is a highly complex process influenced by both intrinsic and extrinsic factors. And furthermore, these studies don't use consistent scoring, variables, standardized data collection, so they don't obviously accommodate things like meta-analyses. Given the current state of the field, I felt it would be useful to try to build a reference database from which models and predictions of PMI could be developed. So these are the goals of the project that were first proposed. So first, we want to build this database. We want to use the data to try to have more robust models and predictions of PMI. And then we want to share the data to advance the field. So I'm going to give updates on each of these goals as we proceed today. Given the interconnectedness created by technology, it easily facilitates collective problem solving using mass collaboration. Mass collaboration projects like Wikipedia, eBird, and the Forensic Data Bank provide powerful ways to tackle complex problems. We know that decomposition is a process impacted by many variables, and it would be nearly impossible for any one person or place to provide the necessary data to make robust predictions and provide a meaningful understanding of the decomposition process. PMI is an area that frustrates many practitioners, and so mass collaboration allows us to work together to improve methods available. The Forensic Data Bank and 4DISC mass collaboration project has been used to tackle sex and ancestry estimation in skeletal remains from the 3,400 cases that have been contributed so far. As another example of a successful mass collaboration project, eBird has hundreds of thousands of volunteers that have reported more than 300 million bird sightings, allowing researchers to model migration patterns and ecological distributions of populations. Data collection projects like these enable researchers to collect data more frequently and in more places than would otherwise be possible. One of the beauties of things like eBird is that it captures work that's already being done, in this case, birding. This feature enables the project to achieve a tremendous scale. The work of observing decomposition characteristics is already happening too. And the goal of this project is to motivate participants through the collective value of knowing that your contribution is helping the greater good while making your job easier. As I began to envision what this project should look like and the application and data collection method to use, I first started with the idea of stages of decomposition. This is a commonly used way of classifying and thinking about remains from fresh, early, advanced, and skeletonized. And I needed uniform definitions for these stages in order to minimize inter-observer error. I found there's a lot of variety in the definitions and characteristics that make up each stage. The notion of classifying decomposition into stages has frequently been used, and it's where I started thinking about this project. But then I began to question, how reproducible are stage information? Do individuals always move through stages in uniform ways, regardless of environmental factors? How well do the characteristics of each stage hang together as an individual passes from one stage to the next? Many of the same questions that have been asked for age indicators like the pubic symphysis. I thought perhaps it would make more sense to begin with the individual variables commonly associated with stages of decomposition so that these fundamental questions could be examined. It's also an important part of the decomposition process to understand the environmental context that's available. Our data survey is built on an ArcGIS platform. It's Survey 123, and so it embeds the georeferenced information about the case in building this repository. By having this geospatial information from ArcGIS, we're able to layer on many other sets of data. For instance, when you drop a pin in the survey, it allows us to go out and ping other weather stations near where the remains are found to gather a number of different weather variables associated with the decomposition process. We're working on looking at other environmental contexts like soil type, slope, bioclimates, soil class, and many other variables that can be used to model the decomposition process because we do have this environmental context. All right, so we have been fairly successful so far in our 18 months of data collection. I'm happy to report that we have collected information on over 2,500 subjects, which far exceeds any existing data set, as shown here with GEO4 at the far end of the graph. These observations include things like longitudinal observations from individuals at outdoor research facilities and retrospective cases from several locations across the US, including indoor cases, urban areas, clothed individuals, and other characteristics that are representative of forensically relevant cases, but not represented at outdoor research facilities. So moving on to goal 2 of the project. So we've built this data repository. We're continuing. 2,500 cases is pretty good, but as my data scientists always tell me, it's not really a large, big data yet. So we need more casework and more geographically representative casework. But I think that we can demonstrate at this point the proof of concept of what this kind of data can provide in looking at and creating predictions of PMI. So these are the results of our initial machine learning models based on this data set. All quantitative methods rely on representative training sets to be able to make reliable predictions of future cases. And this data set allows us to predict PMI with an R-squared of 8.15, which is pretty extraordinary compared to other data methods out there. And this is based on an 80-20 split, a holdout sample. So we hold out 20% of the sample, train the model on 80%, and then test it on that holdout 20%. So it gives us a more robust prediction of our ability to predict future cases based on that information. This machine learning model that we're using is an XGBoost model, which is an ensemble of different decision trees. So we're able to create decision trees based on the observations that we're observing in the characteristics that we're observing in the body. This is very small, I realize, even with my reading glasses on. But in general, if you want to take a closer look, I'm happy to show this to you. Or if you had come to our workshop, you would be able to have seen it then. In general, like with most decision trees, we're starting here at the top of the tree. This says exposed bone with desiccated tissue. And you're going to select either a pathway, either true or false. Do you observe that characteristic on the body? Weathered bone, do you observe that characteristic on the body or not? Skin slippage, exposed bone with moist tissue, and then we come to the end of the tree, a root, and based on this pattern of characteristics, then we're able, I can't read that, to give a predicted PMI of 88 with an 80% confidence interval that I actually really can't read. So that's cool. We haven't been able to visualize this kind of process and set of decision trees based on a large data set before, and so it's exciting to think about the patterns that we're observing using this data-driven approach. We're able to think about what are the different characteristics that are driving these models of decomposition. So in this plot, we're showing if we just include the characteristics of decomposition that were observed on the body, we're able to get a pretty good prediction of PMI. If we include information about insects, we do a little bit better. Information about scavengers, a little bit better. If we add additional information about decomp, insects, scavengers, and temperature, and the deposition type, so whether the body was indoors or outdoors, we do even better. And then you'll notice here, if we add weather data, it makes the model a little bit worse. So we're working on this process of quantifying and thinking about weather. Right now, we have a lot of missing variables in the weather data. So for instance, we have a weather variable about snow. In South Carolina, a lot of the weather stations are missing information about snow. So, and when you run the machine learning model, when you have these missing variables, it takes a hit on the model. So we're thinking about how to better model that weather data. And then if you include all of the variables, we do pretty well too. So that is exciting stuff about ways that we can predict PMI from observations on the body, just making simple observations without requiring any special tools or types of analysis. We're able to do pretty well. Another exciting part when you have these large data sets available is you can do things like Bayesian statistics. So using Bayesian statistics, we're able to model the decomposition process using a data-driven approach. So we can develop models based on expert knowledge. So using prior information from expert knowledge, we're able to model the decomposition process and think about the different variables and how they interact with each other in creating the pattern of decomposition that we observe in the body. So this plot here, these are ROC graphs as the result of these Bayesian statistical models. Again, very small. But each of these labels over here show a characteristic of decomposition that we have documented in the sample. If you have an ROC curve that is on the far side of the graph that goes from 0 to 1, that indicates that this variable is very good at predicting, or it's very good at predicting, and it's highly correlated with PMIs in our sample. The dashed line shows if the prediction is no better than guessing. So many of our variables then do pretty well at modeling the decomposition we observe, and some variables do less well. These models are based on really a lot of input from expert opinion about how we think these different variables correlate with each other and relate to each other and what we think the process of decomposition looks like. So these are just based on our initial sort of impressions of that. Christina and I sort of thought about how we think that decomposition progresses. But these are areas where we can really input and take in other expert information in order to better model decomposition. This plot over here demonstrates that these things could happen. So in this plot, this is using a neural networks approach. So as opposed to taking in expert advice and more specifically modeling decomposition, this uses more of a machine learning or black box approach. So it puts the variables together and thinks about the weights and the priors for each variable in predicting decomposition, and we can plot the data in a similar way. And so we see in this model then that all of the variables do pretty well in predicting PMI and in modeling that decomposition process. So we can start to look at things like the way that body weight relates to mummification, for instance. So we can say, from a data-driven approach, if someone is emaciated, they're likely to mummify early in decomposition, whereas if they're obese, they're more likely to mummify late in the decomposition process. These Bayesian models allow us to do more intentional research design. So places like outdoor research facilities, they can really hone in on what are the things that we know about the decomposition process, what are the things we need more information about, and be able to drive those levers in a more intentional way to document that decomposition process and improve our models for estimating PMI. All right, so another part of this project that is significant is to make the data repository available for people in the field, for researchers to be able to test, refine, and improve models for estimating time since death. Working on this project for the past couple years has furthered my commitment to the importance of open science. New guidance calls on all federal agencies with research and development expenditures to implement policies advancing open access to publications and the underlying data. So this need is broadly recognized. Working on this project has also clarified for me the role open science plays in promoting diversity, equity, and inclusion in scientific discovery. When researchers are dependent on their personal networks to access data, it privileges those who are well-connected and tenured, while disadvantaging those in more tenuous positions outside of these circles. When you create gatekeepers for data, the likelihood of unequal access increases. So this idea, we're continuing to then refine the application to provide information about PMI based on data entered by forensic practitioners in the field. So right now, you can register to become a collaborator, receive the GEO4 survey, and begin to enter data about the bodies that you encounter in the field. We have several ME offices that are using interns to help facilitate this collaboration, so entering cases from retrospective case files. When you submit these cases, you receive an email that lists the data that you entered into the case files. You also will, we're now able to provide that PMI prediction based on that machine learning model that I discussed in order to help to promote and provide people incentives for contributing data. As we have more data, then models continue to improve, and we all make our life easier. So you can, this ongoing data collection is the next step in that process. So we're continuing to recruit collaborators, as I mentioned. If you go to the geo4app.info, you can request access to become a collaborator. And this helps us to better model that uncertainty in the data. So we've designed the survey to be relatively quick and easy to access. It takes just a few minutes to enter this data. We try to provide information about, with as many drop-down boxes and check boxes as possible in order to minimize input error. And we look forward to seeing this data set continue to grow. I think that so far, even with just 2,500 cases and 18 months of data collection, that we're able to demonstrate that this model of examining PMI can be fruitful for future endeavors. So thank you so much for your attention. I look forward to your questions and discussion. And you can contact us. Here's a QR code, or at geo4@clemson.edu.
All right, it looks like we have at least one question in the audience.
This is a naive question. I mean, I assume everybody else in the room knows the answer. But on those graphs you showed at the end of your goal number 2, could you just, I didn't quite understand them. So if you could just tell me, pick any one point on a line and what defined that point, and why did you choose false positives for one of the axes?
Let's see. Can we pull it up again? So we are, I think you want the ROC curves, huh?
Yes.
I might get my collaborator to help me on this. We have also a student in computer science, Noah Nesbitt, who's back there. He's been working on the Bayesian model to help this. So with these ROC curves, help me out, Noah. So as you're developing the Bayesian statistics, the statistical models, you have to build the equation and how you're modeling the different variables. And so this shows you the result of those modeling. So this, for instance, is weathered bone. It has an area under the curve of 0.99. So we know then that if you observe weathered bone in a body, that you're able to make a fairly good prediction of PMI based on the model for that one characteristic. So weathered bone is very helpful, whether rigor mortis is present. So these are all either early, very early stages or very late stages of decomposition, and all of those are, using the model that we're building right now, are very helpful in predicting PMI. But some of these variables, like whether the body is intact but rigor has passed, are not very helpful in predicting the model. Whether those variables are present or absent don't provide a lot of information for improving our model. So, but we can look at each characteristic separately, whereas before we only had stage information, and so we couldn't make those kind of predictive models. Does that help at all? If not, Noah can explain to you Bayesian statistics afterward.
Thank you.
Mm-hmm.
Do you have question?
This is more of a comment than a question, but I think I recognize gathering this data from all these disparate organizations across the country or the world is so difficult. But I think there's an added benefit to this, which is from a crime scene or a search perspective, the actual locations, the precise locations, the kind of environments where bodies are recovered from could be very, very helpful. And also which bodies are buried, which bodies are at the surface. So kudos to you, and I think there's a lot more that could be done with this data.
Yeah, for sure, definitely. Thank you.
We have a question from online. And they say, I see that you are examining open air situations. Are also examining water recovery situations. In my area, fresh water is very rapid.
Yes, so we're collecting data from all kinds of situations. So with the deposition sites, we have mostly a lot of indoor cases right now, which have not been previously gathered. And also, we do have some water data, but the more data that we have, the better we're able to make predictions under those conditions. So that is important.
Wonderful. Thank you again, Katie. Yeah. Fantastic presentation. And we will move on to our next speaker, Amber McGinnis from Florida International University. Oh, gosh.
Thank you for that. So I'm excited today to talk about the research that we've done in generating these fluorescent blowflies as a tool for use in forensic entomology, specifically in looking at validating these PMI estimation methods that we use. So we all know that when you get to a crime scene, a lot of times you'll find insects, and we all know that these can be very useful pieces of evidence for looking at postmortem interval. So specifically, when a person dies, you're typically not colonized prior to death. So we know that using the age of the insects is more than likely the minimum time since death. And that's the most common type of estimation that we do. And the way it's done is by taking this laboratory-generated data from sterile conditions and applying that to crime scene conditions, which can often be very different than those laboratory conditions. So one of the ways, this is just an example here of using, in this case, we use body length there on to predict the age of the insect. There's also ways where people use the stage of the insect as well to predict the age. One of the things, though, that when we talk about scientific testimony, and the Daubert standard says that there should be a known and acceptable error rate. They don't explicitly define what a known and acceptable error rate is, but more than likely this is some type of numerical value, so false positives, false negatives, under realistic conditions. So this means that those models that we use should be validated under crime scene conditions rather than laboratory conditions. However, there is a big problem with that, in that the true age of a maggot must be known from crime scene conditions. And that's very difficult to do because when you have a corpse, how can you know the true age of a maggot? This corpse could potentially have hundreds of thousands of insects. They're going to be mixed ages, mixed species. So even if, let's say, you were to place something out, you were to place a pig, and then you were able to know the exact time that flies lay eggs, it's not practical to track those individuals after hatch. So then how do you recover those same individuals later in order to know the true age? So all you would know from placing something out is that no larvae on that corpse is older than the time of placement, but you wouldn't actually know the true age of any individual on that corpse. So, however, though, if you were able to mark an individual and then kind of do a mark, release, recapture study, then you could be confident of the true age of an insect because you would know exactly when you place those and you could recover those same individuals. However, marking them presents a little bit of a problem because as they transition through their developmental stages, they molt. So they're going to shed their exoskeleton. So any type of external marking would not work in this case. Additionally, any mark that you use can't alter their development rate. If it changes the rate that they develop, then it would make it unsuitable for use in validation studies. So what we propose to do is use transgenic blowflies that are expressing a fluorescent protein. So now you have fluorescent blowflies that you can then go out and you can take those insects back in, you can use a fluorescent light, and you can recover those individuals later. So the goal of the project was to use the piggyback transformation system on a common forensically important blowfly species, Phormia regina, and then to assay this for any fitness effects. We want to know, did this cause any changes in development? So the fly that we chose to use is Phormia regina. It's a very common fly in North American casework. It's found throughout much of the United States, except for South Florida. But we have maintained this fly in colony in our lab. And it is going to be one of the more common species associated with casework. Additionally, there have been 2 species of blowflies that have been, or 3 species that have been transformed previously using this piggyback transformation system, so making fluorescent flies. This represents a separate lineage from all those, and additionally, none of those previously transformed species were used for forensic casework, despite some of those species being forensically important insects. And so this piggyback transformation system that we decided to use is what we call a bipartite plasmid system, in which we have 2 circular plasmids that are injected into the individual. This first one here is what we call the donor plasmid. So it contains our transgene of interest. So in this case, it contains our fluorescent protein, and it's flanked by those inverted repeat sequences there with the blue arrow. Those inverted repeat sequences are also found scattered throughout the genome of the individual. And that's important for how this process works. The second plasmid we use is this helper plasmid, which we call, it produces the enzyme transposase. And basically, it's just a giant pair of scissors that's going to recognize inverted repeat sequences, and it's going to cut anywhere where it sees those sequences. So it's going to cut that transgene out of your plasmid, but it's also going to make cuts throughout the genome of your organism. And that's important because then this cut gene cassette here can kind of be pasted into those cuts in the genomic DNA as a repair mechanism. But when it's pasted in, now you have a new gene function, in this case, fluorescent individuals. So for our study, we tried 2 different donor plasmids. So this first donor plasmid there in A is the species that was, or is the reagents that were used to transform previous blowfly species. And it is controlled by a heat shock promoter. And then it expresses that red protein. The second donor plasmid we tried is much smaller there in B, and it's an artificial eye promoter gene. So in this case, it would only express in the eyes of the individual if it were successful. And then we used the same helper plasmid across all 3 experiments, which is controlled by that same promoter from the donor plasmid marked with A. And so what we do for this process is we take embryos and we line them up, and we're going to inject those plasmids into the posterior end of the embryo. The goal is to create germline transformation, so create something that's heritably passed down from generation to generation, instead of just creating a single generation of fluorescent individuals. So those reagents are then injected into the embryos, and then they're maintained until hatch. Once those embryos hatch, they're transferred onto food and reared to adulthood. Now, once they're reared to adulthood, we want to go ahead and we want to cross them with what we call wild type, or non-injected flies. And so in this case, we're going to cross a fluorescent individual with a non-fluorescent individual and rear those larvae out. Now, if we were successful in germline transformation, once we take those larvae over to the microscope, we will see red fluorescent individuals. And then any of those individuals that screen positive for fluorescence are then repeated with the crossing procedure for a number of generations until eventually we're ready to cross those individuals together to create a homozygous generation. And so what we found was this proved to be very difficult despite the literature being very successful with previous species. So previous species success rate in those crosses ranged anywhere from 11% of those crosses producing fluorescent offspring to 22% of those crosses producing fluorescent offspring. However, with those sets that we tried in that first experiment using the donor and the helper plasmid from the original fly species, we were unsuccessful in obtaining transformation. Once we tried the second plasmid, we were then still unsuccessful using the one where we should have seen fluorescent eyes. However, what we decided to do for the third experiment was we went back to those original plasmids we had tried in experiment 1, and this time we applied a heat shock. Now, according to the literature, this heat shock promoter, a heat shock is not required to produce activity of this gene. It's supposed to have a high level of activity, but we decided just to try it anyway, and you can see there, we were successful in obtaining a single cross that produced fluorescent offspring. And so eventually we did find 8 fluorescent individuals from a single injected male. So there on the left, you can see we have a wild type or a non-injected individual, and on the right, we do have our fluorescent transgenic offspring. And so eventually we did cross those individuals together to produce homozygous individuals that produce a different phenotype. So you're able to tell a heterozygous individual, so containing 1 copy of the gene, from a homozygous individual containing 2 copies of the gene. So there on the left, again, we have wild type. In the middle, we have our heterozygote, which has a slight pink coloration to it. But then there on the left, we have our homozygotes. And it doesn't show up quite as well up there, but they have a very distinct pink coloration, even under white light. And then once you get them under the fluorescent light, we do have a much brighter fluorescence in the homozygotes compared to the heterozygotes. So this is potential to even use. You have 2 different phenotypes from the same line that can be differentiated. So, and then across all the different stages, we found that we have fluorescence ranging from the eggs all the way up to the adults. So even those adults that are very darkly colored, we're able to find areas of fluorescence across everybody. And even through that pupil casing, we're able to see fluorescence, which is cool for imaging like development models and stuff like that. So the next thing we wanted to know is, where did this insertion land? So remember, those inverted repeat sequences, they're scattered throughout the genome. So this could end up in an intergenic region between genes, or it could end up smack dab in the middle of a coding region, which would then be likely to cause fitness issues. So we wanted to know, where did this happen? So we did short read Illumina sequencing on all of those original adults that were produced, and then we did an assembly of all 6 of those genomes. And what we found was every individual had only a single insertion. So we did a blast of the fluorescent protein coding sequence against all 6 of the assemblies, and everybody only contained 1 insertion. And we found that everybody contained the same single insertion. So all of those flies were identical. And then the next thing we wanted to know, none of this tells us whether or not we're in a coding versus non-coding region. And so we took transcript data from wild-type adults and aligned that with our genome assembly. So we took all the genes in adults that are expressed, aligned that, and looked at our region of insertion to see, are there any genes that align there? And what we found is there's no genes that align in the region, and when you take a 2 kb region and you blast that, there's no significant blast results to any known protein coding sequence as well. So this does suggest that we landed in an intergenic region of the species, so more than likely, hopefully, we hope that there's no fitness effects that result from this. However, though, in order for this to be useful, we need to be sure that there is no effect on the development rate. So the next thing we did is we looked at this. And remember back, so future work for this is going to involve using those wild-type models that are created in the lab and applying those models to these transgenic larvae in the field or under a crime scene conditions. So we want to examine how well do those wild-type models work in a nice controlled laboratory setting. If it works in a laboratory setting, then it's more than likely making them ideal candidates for field conditions. And so we took a bunch of eggs from all three of those genotypes. So we had wild-type heterozygotes and homozygotes. And we had a number of sampling times. So we sampled at 18 time points throughout their larval instars, and then continued sampling throughout the pupal and adult stages until everybody had reached the adult stage. And then when everybody was sampled, we were looking for a couple different things. So we were looking for first the stage of the insect. So we were looking here for L1s, L2s, or L3s, so first, second, or third instars. And then in pupal development, we're looking for the presence or absence of those respiratory horns. So the respiratory horns appeared during the second half of pupal development. So we were able to break that stage in half. And so using that data, we used the wild type data that was collected to make a 95% statistical age prediction table. And so what we're looking at is then we apply that 95% statistical table to the heterozygotes and to the homozygotes, and we look how often does that model include the true age of the transgenic flies. So in an ideal situation, we would want this to be 95% or better. And so this is what the wild-type model looks like there. So the unshaded cells represent the 95% confidence prediction per stage. So you have stage here running across the top. So for an L2, for a second instar individual, your 95% confidence prediction would be anywhere between 36 and 72 hours. And so we then took this model and directly laid it over tables comparing to the heterozygotes and the homozygotes. And what we found was that the accuracy of this prediction was greater than 99% for both transgenic genotypes. And so here you can see what it looks like for the heterozygotes with those blue shaded cells representing true ages of a heterozygote that were not included in the model, so they were missed predictions. But even with those few missed predictions, the capture rate was 99.7%. So if you were to use a wild type model to predict the age of a heterozygote, it would include the true age 99.7% of the time. And then for your homozygotes, it's going to include the true age 99.8% of the time. So from this, we can conclude that it does not appear that this insertion then caused any changes in the development rate of wild type versus heterozygotes or homozygotes. And so while this work was more difficult than anticipated, we were successful in creating a single individual or a single line that is transgenic. And because it is in an intergenic region, it's less likely to cause any type of both developmental or other fitness effects. So this is preliminary work for the fitness data. We are still analyzing, we are comparing length and width as well to make a multivariate model to help refine this prediction. So we're in the process of finishing up those numbers there, but preliminary fitness based on stage does not show any change in development rate. So then this would be possible then to take these age estimation models that we use in forensics and validate them under crime scene conditions, which to my knowledge has not been performed in the field. So I want to acknowledge everybody that's assisted on the project and of course the funding, NIH, and of course NIJ for this grant funding that made this possible. And with that, I will take any questions.
Thank you so much. Always an excellent presentation and always give me like an idea for questions to ask, but I'm only going to ask 1.5. One is, do you think the preservation method or diet or whether or not your samples are dead or have been dead for days might have an effect on the fluorescence that you're appreciating, considering also that you're looking at brighter versus less bright individuals. And the other half of the question is if you could suggest or briefly mention some practical applications that this might have in the postmortem estimations.
For sure. So I haven't looked at the effect of preservation, but obviously things like UV light are going to degrade the fluorescent protein, especially once the insects are preserved. While they're alive, they're continuing to produce more of the protein, so it's not a problem. But once you preserve an individual, it doesn't appear so far that our preservations have done anything, and then it's even still been under dark conditions preserved in ethanol, we've had fluorescence of the individuals for months after they've been preserved. But obviously exposure to any type of UV light would then degrade that. So, and then the second half of your question is we want to test how these models perform for PMI. So we don't know when we make a PMI estimation from insects, we don't have a confidence that's associated with that. We can't, say that, yes, we're 95% confident in our prediction or anything. So this would be the major application, is testing those models to see how often it includes the true answer to kind of give that. But then there's other things that could be done other than just PMI in terms of looking at the behavior of these individuals. You could now track individuals throughout a corpse. So we know that adult flies tend to move around in sibling groups. But do larvae then tend to congregate in sibling groups? We could look at thermoregulation behavior and things like that as well with this.
Any other questions for Amber? Okay, thank you again, Amber. Fascinating. All right, and our next presenter is Giovanna Vidali from University of Tennessee at Knoxville.
Thank you. Joanne and I are very excited to be presenting here today, and I will be introducing our project and some preliminary data. This was a 3-year project that started January of 2020. And unfortunately, we face a lot of COVID delays because we rely on body donations. And it concluded this past December, but we have requested a no-cost extension to wrap up the final phases of our project. During this presentation, you will be seeing images of real human bodies in various stages of burning. These are bodies from the Forensic Anthropology Center Body Donation Program. The individuals are self-donated and have also indicated that they would be willing to be used for trauma research, including thermal trauma. But out of respect of the donors themselves and this research, I do ask that no images or pictures or anything are taken of the bodies. As you can see, the number of fire fatalities has increased in the last decade, spurring the need for this whole body burn research. Most of the fire fatalities occur either in house fires or car fires. And one of the questions that we get asked often is, can you tell the temperature of the fire based on the destruction to the body? And the destruction that we see, or the impact on the body from fire, is really due to these two factors: the temperature of the fire and the duration of the fire. So a house fire burns at a lower temperature than a car fire, but if it burns longer, we will see the same amount of destruction. So you could have a lower temperature fire burn for longer than a high temperature fire, and it'll cause the same amount of destruction. So we do need to always keep these two factors in mind of temperature and duration. In bone or in bodies, we generally see about 5 stages of impact from fire, starting with charring, singeing, and blistering of the skin. As the fire starts, the skin starts to dehydrate, the inner tissues start to swell, so we start seeing some skin splittage and fat exposure. As the fire continues or the temperature goes up, we start seeing partial cremation and charring of the distal appendages. And again, if there's continuation of the fire, we start seeing some variation, maybe some incomplete cremation. The extremities are disarticulated. The body's not quite whole anymore. And then as the fire continues, there's no soft tissue left, and the bone is incredibly fragile. So in that last image, where you see that the bone is white, that's calcined, and it can easily fragment either during recovery or even during transportation. So we do see these additional fragmentation, not only due to the fire, but also due to recovery and transport. There are some documented thermal fractures that we really only see as a result of the fire on bone. And you can see these here, longitudinal, thumbnail, that sort of mosaic pattern of the patina, or delamination fractures. But in this research, we're not only interested in the thermal fractures, we're also interested in the identification of blunt force fractures in burned bone. So for blunt force on long bones, you might see some transverse or compound fracture patterns. On the cranium, you can see concentric or stellate and linear fractures. And so our research questions here were really to help the practitioner, whether that is a forensic anthropologist like myself or a medical examiner, to see if blunt force fractures, perimortem blunt force fractures, are still visible after a fatal fire. So did something happen to that body prior to the fatal fire event? And as this part of this research, we have two aims. One is, can you still see perimortem blunt force fractures after a fire? And how confident are you that that is due to blunt force fracture or blunt force and not fire? And then the second aim is really to provide the best practices to our colleagues to successfully differentiate between blunt force and thermal trauma. In fact, one of the cases that my colleague Joanne Devlin is currently working on is a fatal fire scene where she was asked, did something happen to this individual before the fire? So we see a need for this. The project is in 3 phases. So we have completed phase 1, the data collection phase, and that's what I will be talking about today. We have begun phase 2, and I will be introducing that as well. And we will use the data from phase 2 to inform phase 3. And that is, again, the best practices to develop a scoring procedure. Or ideally, like Katie also presented, a decision tree to see how confident we are in saying this is definitely blunt force trauma. And then we will validate that scoring procedure with our colleagues. So to carry out phase 1, the first thing we needed was a way to impact the bodies consistently, and in a way that we could also record the amount of force, control the amount of force, control the duration of impact, and the location of impact. So we worked with undergraduate students from the engineering department to design this impact table. And they actually presented it as a senior engineering school senior showcase, and they won first place for it. Very proud of them. So here you see Joanne and I. So this is the impact table. This whole thing is called the gantry, and it is movable. It moves the whole length of the table, so the body can be impacted anywhere from head to foot. This here also moves up and down, so we can control the distance to impact. This here moves left and right, so we can control where on the body. It is pneumatically powered, so here we see an air tank connected to a larger compressor. And we can control the force by changing the PSI. The engineers had looked at different methods. Previous researchers who have done blunt force trauma research have used something like a drop tower, but we needed enough force to successfully break pretty sturdy bones like the tibia. We also had a load cell that recorded the amount of force that we used. So here we see on the y-axis is the amount of force in pounds. So think about this as like an inverse stress-strain curve. Here we have the amount of force in pounds, so we can input it in PSI, but the output is in pounds. It's in negative because these were all compressive forces. Here we see the time. The load cell recorded the time in milliseconds. And again, we could record the duration of impact. For the long bones, it was 1.3 seconds, and then for the cranium, longer. So we have all the data on the force required to break these elements, and this is for a tibia. And here, that's the load cell right there. And that's the impact, one of the impact heads, that's a pig skull. We had 3 impact heads, and for all of the impacts for this research, we used either the square or the rectangular one. So we impacted the bodies in 3 locations. One was on the cranium, right above the ear on the temporal. The other one, another location was lower arm, so the radius and ulna on one side, and the other side served as the control. The lower leg, the tibia fibula on one side, and then again the other side served as the control. We enrolled 16 donors who fit our research parameters in this research. So that would be they consented to trauma research, but also they weren't autopsied, for example, which leaves 32 limbs for comparative purposes. And here you can see what we fractured. And so remember, the other side serves as the control. We fractured 10 crania, and then we had 6 as a control. Because what we're trying to see here is, can we see blunt force fractures after an event, and is there a difference in patterning between blunt force and thermal trauma? We took x-rays at 3 points during this research. First, before impact, we x-rayed the cranium, both sides of the radius and ulna, both sides of the lower leg and the thorax. This was to confirm no surgical plates, no joint replacements in those areas, and the thorax for no pacemakers or medical pumps. Then we also x-rayed again after impact. So now we know the type of fracture and the location of fracture. So here on the cranium, you can see that concentric fracture with some radiating lines. Here on the radius and ulna, the transverse. And then here on the tibia, we have a butterfly fracture and a common unit fracture on that proximal fibula. Following impact, we proceeded with the thermal trauma part of the project. So we built 2 forensic pyres out on property owned by the University of Tennessee Institute of Agriculture. The forensic pyres are 3-sided, and we had 2 so we can conduct 2 burns at approximately the same time. The fire source was locally sourced wood, basically from the trees that we felled from that property. To start the fire, we used drip torch and fire starter pellets. We did not use any accelerant on the body. All of these accelerants were on the wood itself. Here is another image of the setup. So we've got that 3-sided cinder block structure with the wood underneath. These two wires, aluminum-wrapped wires that you see extending, are thermocouple wires that were recording the temperature of the body during the fire and the temperature of the fire itself. We burn the bodies to approximately a stage Glassman-Crowe 3 or Pope et al. 4, where there is extremity disarticulation and some bone fracturing. We put out the fire, we would intentionally put out the fire either by raking out, well, both by raking out the hot coals from underneath and also cooling down the pyre. We didn't want to water the remains themselves, as that could cause further fracturing. We collected multiple data points during this process. They're not all related to the questions specifically related to this project, but we see lots of future projects coming from this. So this is the thermocouple, the recorder. We have these 4 wires extending because we had 2 burns going at the same time, so 2 for each body. So here, channel 1 was the temperature of the body, and channel 2, the temperature of the fire itself. We also took individual FLIR images. We recorded the amount it took for each burn, for each donor. These are the data for the first 8 burns. On average, it took about 2 hours per burn. And we also recorded the amount of wood we used, just to have an idea of the fuel load that was required. We also set up a camera to take an image, to automatically take a picture every minute. So for the 2 hours, we have 120 or more images taken every minute of the body while throughout the whole burning process, so we can get an idea of the body movement and the process. So in the next dozen images, you will see some images of the body burning. And just take note of the process. You will see some skin charring and skin splitting with fire exposure. Also a lot of limb movement, both in the arms and the legs. It's pretty continuous how the body continues to move. You'll start seeing the right leg going up as the fire continues. So once we finished with the thermal process, we had to recover. So in some cases, as you can see here, there was extensive bone fragmentation, so we would recover the bones and wrap them in aluminum pouches. This helps keep the bones, somewhat stable and secure. Also, we could label the aluminum pouches, so we could say this was left tibia fibula, which then makes the lab analysis easier. And you could also X-ray through those aluminum pouches, or we would put foil on areas that were delicate. So back at the lab, we X-rayed again. We prepared the remains for processing, and we also took DNA samples for another NIJ grant that's called Optimizing DNA Analysis from Burned Remains, and they're presenting later this week. Tomorrow, we have a poster presentation on the challenges in processing burned remains, such as those. So once the remains were all cleaned, we have started analysis on one bone. Right now, we're focusing on the tibia. We are collecting all the data in a Google form, so it's just direct data analysis. And we are using the classification system from the Journal of Orthopedic Trauma, just so we can systematically and consistently record where we're seeing trauma and what type of trauma. So all the long bones are divided into 5 segments under this system. The diaphysis is in 3 segments, the proximal, middle, and distal third. And then we also have the proximal end and the distal end. So for each long bone, or for each tibia in this case, we are asking the same question of 5 different parts of the bone. So the first question is this portion an isolated segment? So here you can see this is the proximal end, and it's an isolated segment. It's not attached to any part of the diaphysis. This one here, the proximal end, is not isolated, but the distal end is. And we started asking this question because we started seeing a pattern that consistently the proximal end seems to be isolated. So we just are wanting to record what kind of patterns are we seeing. Now we know where we impacted the bones. So this isn't, as we're doing data collection, we're not at that point where we're just trying to see if the blunt force is visible. We want to record if we did impact it there and if we do see it. So here, for example, that's the proximal end, not impacted by blunt force trauma, but yes, an isolated portion. This is the proximal third diaphysis, not impacted by blunt force trauma, but yes, there are radiating blunt force fractures. And then for this middle third of the diaphysis, yes, impacted by blunt force trauma. We then use the type of blunt force trauma from, again, this classification compendium. And the one that we are seeing most often in the tibia is a fragmentary wedge fracture. So that code, which is probably hard to see, is 42B3. And then if it's in the middle third, it would be 42B3B. And so now we're just, again, consistently recording what type of blunt force trauma we are seeing. We also record the color of bone. Bone changes color throughout the burning process, and the grayer or whiter it is, the more fragile it is. And so for us, this color gives us an idea of the fragility and the ease in which it could fragment, again, either during recovery or transportation. And then we also record if that area displayed post-fire breaks. So is that break that we're seeing due to pre-fire, perimortem, during the fire, or after the fire? So a couple of interesting things that we are seeing so far. Oh, sorry, one other part is what type of thermal fractures are we seeing? So here we see thumbnail fractures, longitudinal and step, and a superficial heat line fracture. The most common one that we are seeing are the longitudinal thermal fractures. So a couple of points of interest as we're going through this. So this is a distal tibia. These are 2 separate fragments that we have refitted, but you can see that one part has burned for longer, it's a different color, than this unburned part. So one may look at this and say, okay, well, there was a fracture prior to the fire, which caused this area to be exposed to the fire longer than here. But we know we didn't fracture this part. We know that there was no perimortem fracture. So what we're seeing must be due to normal part of the burning process that happens. Here is another 2 others. These are from the same individual. You can see that they are fractured in the same location. One is due to blunt force, one is not due to blunt force. One is due just to thermal fire. So this is another thing. What is causing the bones to break in these locations? And why? And so we're going to start looking more, a lot more at that anatomy and what happens during a fire, which is where those images are going to be very helpful as well. Here's another instance where we see somewhat of an oblique fracture, and again, an area of differential burning, but not due to blunt force trauma. This is just a result of the fire process. So we are going to complete data collection on all the bones, and again, once we take all of these data, a lot of it observational, but we are working on making it, coding it, and making it systematic, creating a decision tree to see if that fracture is due to blunt force trauma, thermal, or another aspect, such as recovery or transportation, and then test this with our colleagues. The goal is really to provide our colleagues with a method that has an error rate to say, yes, this is definitively blunt force trauma or not in a fatal fire death. So that's it, but before I go, there is a PSA. Please check your smoke detector batteries.
Looks like we have a question from the audience.
Does having a pacemaker alter bone morphology? Why did you exclude such individuals?
Because it can explode during the fire, and so we need to remove it if they do.
Gotcha.
Oh, we have a virtual question.
We have an online question. Have you already, or do you plan to X-ray or otherwise scan the impacted and thermally degraded bones to compare to the pre-thermal event bones?
So we X-rayed the thermally impacted bones, but not all the bones, so just the radius ulna or tibia, fibula, and cranium.
Another question from online. Do you compare the location and pattern of fractures post-impact and thermal event to antemortem records to identify weak points, healed fractures, pathology, etc.?
That's a really good question. We X-rayed to see if there were any antemortem fractures. In fact, we excluded one individual because we could see a callus formation there, but we have not looked at the health records or previous pathologies of that person or any of the individuals.
Wonderful. Thank you again.
Thank you.
And now our last speaker before lunch is Angela Hardin from Ohio State University.
Thank you. So I'm going to be talking today about the development of the Forensic Anthropology Skeletal Trauma Database, or the FAST database. Before I get started, there are a lot of people that contribute to this project, from undergrads to faculty, and so just thank you to everybody that has put effort into this project. So standard disclosure, nothing to disclose. So this research is part of an ongoing project which conducts controlled experimental bone trauma research utilizing a multidisciplinary approach through collaborations with forensic anthropologists and biomedical engineers. This research focuses on fracture mechanics to improve the validity of skeletal trauma analysis and interpretation through precise, accurate, and repeatable analytical methods. So the Forensic Anthropology Skeletal Trauma, or FAST database, is not simply the dissemination of an extensive amount of data from an ongoing research project. This database provides data from experimental testing parameters to fracture characteristics, from skeletal trauma with known loading parameters to allow users exposure to experimental skeletal trauma cases to help develop and improve education and to allow for comparison to forensic casework. So the largest obstacle for the field of forensic anthropology is exposure to trauma analysis. Few researchers get quality hands-on training with trauma cases, and even fewer have experience with cases involving known loading conditions and injury mechanisms. So improvement in skeletal trauma analyses and interpretations is dependent on dissemination in a user-friendly format that allows for training and education and supports forensic professionals in practice. The everything included currently in FAST is loaded in this loading condition. So we'll eventually have 100 tibiae, and they're dynamically impacted at midshaft in a 4-point bending testing scenario. So as we can see in this video, we're looking at the posterior surface of the tibia, and in this, the impact is in a lateral to medial direction. And so this project includes 6 test series. So we have our tibiae impacted at 6 meters per second in a lateral medial direction. We'll also do a posterior anterior direction. And then we'll have tibiae that are impacted at 2 meters per second in lateral medial and posterior anterior directions. We'll then have tibiae that are impacted at 6 meters per second with an axial preload, so as if somebody were standing on that leg at the time of impact. And then we'll test an additional series of fleshed legs to look at differences between these different conditions. So today, I'm really just going to walk you through what is FAST, how to access it, and then what you can do within that database. So to request access for FAST, you're going to go to the Injury Biomechanics Research Center website, and so that's ibrc.osu.edu. And this brings you to our homepage, and so then you will want to, they said push it hard, go to the FAST site. So you'll click on that tab, and that'll bring up the FAST website within the IBRC page. And so this is the top header of the FAST page within the IBRC website. And there's that link as well if you just want to head directly to that page. So you scroll down the page, and you'll see it says to request access to the FAST database, click here. So you could go ahead and click there. And it'll bring up our database access request form. So you fill out the simple form, it's first name, last name, title, and email. And then you're going to click on the next page. And so that brings up the data use agreement. So you'll, of course, read through everything, and then you're going to add your signature at the bottom, and then you're going to click submit. And this will send this through our system, and I'll get an email. And it says, so then you get the confirmation, so thank you for requesting an account. And then you'll be notified by email soon about your request. So you'll get 2 follow-up emails. One will tell you that you have been given access to the FAST database, and it'll give you a link in that email to access FAST. The other email will give you a link to set up your password. So you need to set up your password we use the program REDCap, and then you can go ahead and access FAST. If you are like me, that email will get lost in all of your emails, and you'll never find it again. So you can go back to the IBRC website right there and go to the FAST page. And then down here is to access the FAST database. Click here. That will bring you to the REDCap login. So once you log in, you'll need to go over and click My Projects. And then once you do that, the list of all your projects, if you had more than one Red Cat project, would show up here. And then you just click on that Forensic Anthropology Skeletal Trauma database. This will bring you to the home page of the database. And so here we provide some information about the funding for this research, about why this research is important. And then there's a brief summary of the different types of data that can be found within the database. And then we are definitely interested in feedback. We want to know if this is working for you guys, if there are additional elements you'd like to see, or if there's additional data that would be helpful for you in education and training in comparison to casework. So please let us know how it works or if there's issues. So to search fast, there's two different ways that you can search the database, and those are listed on the left. There's the data exports, reports, and stats, and then search dashboard. So we'll start with the search dashboard, and that brings up a new page. And you have the option to select a search field and then search text. So for my search field, I put in sex, and for my text, I put in female. You then click on search, and that brings up the records that were any record that was female within the database. So within each row, you can then go ahead and click on the Open tab, and that will open each record individually. So search dashboard is good if you just want a quick search of available records. But it is limited search fields, such as age, sex, and stature. And then it's a limited search text available within each of those search fields. And these searches cannot be saved. So once you run it once, it's not saved automatically where you could go back in and look at that report again. So alternatively, you can do a different type of search, more of a query, to look, and you're going to use this data exports, reports, and stats. And that'll bring up this page here. And you'll click on Create New Report. And then you can enter a name of report, and then a description is optional. But if you're doing multiple queries and these reports are saved, a description might be helpful for you to know exactly what you queried to get those results and records. So I just put in blunt force tibia down here. So then you'll scroll down, and these go by steps. So step 2 is fields that you want to include in your report. So I said, I want to know the record ID. I want to know the age of the individual, the sex. I want to know the side of the element, the mechanism of trauma, and the loading direction. Then step 3 is you can do optional filters. So I went ahead, okay, I want to limit this to females. I want to limit it to left tibia only. I only want to look at blunt force trauma. And I want to see things specifically that are in a lateral medial direction, because I'm looking for something to compare back to what my case is. Then you'll click Save Report, and it'll say, your report has been saved. And then you click View Report. Then this comes up. So this is your results of your report. It'll have the report name, the description, and then it'll tell you how many number of results were returned within that query and out of how many total number of records. And then below are all of your records that were returned for that query. So through here, you go in and click on the actual record ID, and that brings up your record. So we'll start with the data that are included at the top of the page. So for every record, you have subject level data. So you have age, sex, and stature of the individual. Then you have experimental testing data, so the setup of the test. So mechanism of trauma, blunt force, loading direction, lateral to medial, because that's what I requested, and then the loading rate. So in this case, it's 6 meters per second. Then we have an area for injury data. We haven't input these data yet for the tibia. That will be coming up. But we'll also be looking at doing incorporating other components like other bones, but also whole body examples and cases. So we'll want to know that AIS is the abbreviated injury scale, and so we can assign scores to the injuries so that we can compare across cases. So I could look at all of my cases that were the blunt force, lateral, medial, 6 meters per second, and see if there's any variation in that injury scale. Then we have our element level data. So it tells me that I'm looking at the tibia, I'm looking at the left tibia, and then we input the measurements that were taken prior to testing. So all those are available for comparison as well. Then down below, we have the project that this came from, because there will be multiple projects involved, and you might want to get more information about that project and the objectives of it. So the project's listed there. And then this will also be added in the near future, is that biomechanical data. So what was the force at fracture? What was the force at yield? What was the displacement? Things that we want to gather about the test and about the response. And then down at the bottom is our fracture data. So we also use AOOTA, and thank you for describing that so well before. And so we report the number of fractures, the classification, like on a broad scale, was it complete or incomplete, and then the type, group, and then a broad location and a specific location. So for our specific location, we designate that as a percentage along the length of the bone. So this fracture was at the 44% site of this tibia. Then you keep scrolling down the page and it'll tell you what image data are available for this record. So for this record, we have a pre-test X-ray and AP, pre-test X-ray lateral medial, post-test X-ray, and in both directions as well. So here's an example of what you'll see when you look at the pre-test X-rays. So on the left is the AP, and on the right is the lateral medial. And then here are examples of those post-test X-rays that are also available in both directions. So these include those squares are how the bone is fixed and held in place for the testing, and those are included in all the X-rays. And then here's our other direction. So the data report is better in the sense of if you're looking to get some specific information and you know what you're looking for and you need to go through and filter and query in that manner. And it has unlimited fields to include in your report, so you don't have to limit it to one or two things. And then there's the advanced filter options to help you get exactly what you're looking for. You have the ability to order results, so maybe within age you wanted to look at somebody at younger and older. You have the ability to order those results. And then these reports can be saved within the REDCap FAST database. I'm not sure how many will keep on the actual website at this time, because really it's like 5 people using it now since we just started it. So I don't know how popular that feature will be, and we'll have to figure out if we're going to keep them out there forever or for a limited time. So the future of FAST. Right now, of course, it's only limited to tibia, but we do have plans to incorporate additional elements. So these will have ulna, radius, and ribs, and these are from previous and ongoing projects going on in the Injury Biomechanics Research Center. So we'll add in those elements and those testing conditions. We also will be incorporating 3D models reconstructed, like you see on the right. This is to help you get a better understanding of how it actually looks in the 3 dimensions, because it's hard sometimes when you look at an x-ray to see how that fracture actually looks on a bone. The goal is to have these incorporated where it's not just spinning reconstruction, that it's actually the user can manipulate how they want to view that model. We have fracture surface images of tibiae tested so far. Those will then be uploaded into FAST. There'll be 2 images of each surface, one that's not annotated and then one that is annotated with some of those fractographic features, if you're interested in education and training on that method. And then, as I say, we also will be including whole body experimental cases. And so it'll be something where you'd have to go in and select if you want to look at single elements or whole bodies, because that would bring up each body for comparison to looking at different loading situations. And then some advanced statistical output. So you can say, okay, doing my analysis and my interpretation, and I'm saying that this occurred in a lateral medial direction. What's the frequency of that happening in this known loading condition? And like I said, we really want input. We want to know what you like, what you don't like. And so on that home page is the blue button in the left-hand side, and it says Contact a FAST Administrator, and that will just send me an email. You can let me know things that you would be helpful for future analyses, different elements that you're interested in seeing, different loading conditions. So I'd just like to thank the anatomical donors of The Ohio State University, without whom this research would not be possible, the National Institute of Justice for funding this research, and all the faculty, staff, and students in the Injury Biomechanics Research Center for all their support, PIs, and other investigators on this project. Thank you.
Do we have any questions for Angela? Any virtual questions? You were so comprehensive.
Hello. Hi. I have a question.
Oh, wonderful.
Here.
Hi, Christina.
Hi. So once we request access, how long would we have until we get the access?
I have no work-life balance, so I will see the email immediately and just go in and approve it. Okay. It then gets sent to like an administrator, and I think he gets it done within several days.
Yeah, that's great. Because I would love to use this as a resource for my students, especially, images of trauma. We have a few bone casts, but not a lot. Yeah. So I would love to do that.
Yeah, please.
Okay. Thank you.
I saw that the tibiae were not fleshed. Do you expect there to be a difference in sort of force that's required, or fracture patterning, fleshed versus not fleshed?
We're seeing similar fractures in these tests that we do in cases from medical examiners. So I'm not sure that the fracture classifications or patterns will change. I think the flesh will, of course, dissipate some of that force, but 6 meters per second, I don't think that'll stop it or slow down too much.
Okay, well, thank you again. And let's give one more round of applause to all of our speakers. So a couple quick announcements before we break. So we will resume back at 1:25 with our next session on seized drugs and toxicology. But in the meantime, I want to let you know, to your all right, my left, is our poster session is now open for self-guided, and then from 5 to 6:30 this evening, the researchers will be there as well. So feel free to browse the poster session, and then in the meantime, and then from 5 to 6:30 as well. Thank you.